Hello Year 11. In this video I'm going to talk through some content that could come up in the exam on Tuesday in the biology paper 1, some predicted ideas and things that might come up. Okay, so number one is bacteria. Lots of things that could be associated with this. So we'll start with basic cell structure in terms of a bacterial cell. We need to know that bacterial cells are prokaryotic cells, and this means that they don't have a nucleus. That means the DNA is free inside of the cytoplasm. I'm going to talk you through the different structures that we have now. So around the outside of our cell, we have a cell membrane that of course allows things in and out of our cell. We also have a cell wall which surrounds our cell membrane. We have a cytoplasm. This is where chemical reactions will be taking place. And we have these loops of DNA that are not inside a nucleus. And we also have these things here called flagella. These are tail-like structures which are not always found but often found on our bacteria to allow them to move. Bacterial cells are unicellular, and that means they are only one cell big. Here's another example of a diagram of a bacterial cell, just with a slight difference here. So we've still got our cell wall there, we've got our cytoplasm, we've got our loop of DNA, but we've also got a plasmid, which is another way of storing the DNA. It's simply a ring of DNA. So bacteria multiply quite regularly. This is their way of reproducing. So bacteria multiply by a type of cell division called binary fission. They can't do a cell division by mitosis because they just don't have a nucleus. And this can happen as often as once every 20 minutes, but that depends on the nutrient availability and also the temperature. Bacteria can be grown in a nutrient broth solution, or we could grow them as colonies on an agar gel plate, and the agar would act as a source of nutrients. We need to know that to actually look at results of investigations, thinking about trying to investigate disinfectants and antibiotics, we need to have uncontaminated cultures of our microorganisms. And we're going to talk about something called aseptic technique and how that can be achieved in the next slide. So this is a required practical where we would be given some agar plates with some bacteria that had been growing on there, and we would then place some little discs of paper which had been soaked in different disinfectants or antibiotics. Now, when you're doing this, you need to be following this idea of an aseptic technique, and this is where you are ensuring that you are only growing one type of bacteria on your plate. So to do that, you're going to ensure that your petri dishes and the culture media are sterilised before use. And that means you're not going to grow any nasty bacteria, you're only going to grow the bacteria that you are interested in. We also talk about inoculating loops, and you can see there's a picture of one at the bottom of the slide. These are used to transfer the microorganisms to your media. So from a little bottle into your agar gel, and you sterilise these by passing them through a flame, and that makes sure that there's no bacteria sitting on there that you could transfer over to the agar plate. We're also going to make sure that the lid of our petri dish is secured, making sure that there's some tape around that, and that means that no airborne bacteria can enter our agar plate and start to grow. We're also going to store it upside down, and that ensures that any condensation that would have been going onto the lid doesn't drip down onto our agar gel. And in school laboratories, we're going to incubate at about 25 degrees Celsius. The other thing that you might be asked about in terms of bacteria is around bacterial diseases. There are two that you need to know about. The first is Salmonella. Salmonella is spread by bacteria that is ingested in food or on food that has been prepared in unhygienic conditions. Now, bacteria cause cell damage by releasing toxins, and Salmonella bacteria can cause the following symptoms. They can cause fever, abdominal cramps, vomiting and diarrhea. Now, in order to try and prevent the spread of Salmonella, this typically comes through poultry products, so this is from typically chickens. We can actually vaccinate the poultry against Salmonella, so the bacteria cannot be in that meat or in those products, and that means that it can't then spread to humans. So that's one way of preventing the spread. The second bacterial disease is gonorrhea. This is a sexually transmitted disease. And gonorrhea, again, is caused by bacteria, which releases toxins, and this can cause the following symptoms. So thick, yellow or green discharge from the vagina or penis, and pain on urinating. Now, because it's a bacteria, we can treat it with antibiotics. In fact, it was really easily treated before with the antibiotic penicillin, but unfortunately we've got resistant strains that have appeared, so we have to be careful about which antibiotic to use to treat a bacterial disease. And we can talk about how to prevent the spread of this bacterial disease, and that is mainly through the barrier contraceptive methods. And this means that you don't have sexual contact during sexual intercourse, which means the bacteria cannot spread. Okay, next up is gas exchange. Okay, so just watching this animation, we can see particles moving. Now, at this moment in time, you can see that these particles have reached something called an equilibrium, and that's where the concentration on either side of this line that we see down the middle is equal. Now, in reality, these particles would continue to move. We just saw a snapshot of time. And this process that we've just seen is called diffusion. And what we have down the middle of the slide is a cell membrane. So substances may move into and out of cells across our cell membranes via this process of diffusion. And diffusion is the spreading out of particles, which results in the net movement of particles from a higher concentration to a lower concentration. When we talk about a net movement, we're thinking about an overall movement that is occurring of our particles. So diffusion is really important in gas exchange, and that's our next thing that we're thinking about. We're thinking about exchange of materials. Now, to understand this, we need to understand that diffusion can be affected by a number of factors. First one being the difference in concentrations. We call this the concentration gradient. If there's a really steep concentration gradient, that would mean that there is a big difference in concentration between two regions, and our rate of diffusion would be much greater. Remember, diffusion is a passive process, and that means it does not require any energy. Second factor is the temperature, and this relates to the particles having more energy. If a substance has more energy, then therefore it will have a higher temperature, and this means your particles will spread out quicker. And the third factor is the surface area of our membrane. If you have a really large surface area, it means that there will be more space where the diffusion can take place. So gas exchange is just one example of how materials get exchanged in biological systems. You need to know about a few different examples and how these examples are adapted for efficient exchange of materials. So the first one I'm going to talk about is the small intestine in mammals. So if we look at this, here's an image that was from an exam. This is what we call villi. These are small folds in the wall of your intestine. We've also got lungs in mammals as a place where gas exchange would take place, and this specifically takes place at the alveoli, which are tiny little air sacs where the gases will be moving from the air in your lungs into your blood. We also have gills in fish, very similar in the sense of a lung, but this time we're obviously in water, so the particles will be moving from the water and into the blood. We also have roots in plants, where we can think about the root hair cells having a really large surface area to allow more things to move into our cells of our root, and we can also think about the leaves in plants. And this is where we can think about our structure of our plant and the idea that we're trying to get as much carbon dioxide into the leaf as possible. So we're going to talk about now how all these places, these exchange surfaces, are adapted for efficient exchange of materials. Okay, so in all of these examples, the villi, the lungs, the gills in fish, the roots and the leaves, we can say that there is a large surface area, and that means there is more space for diffusion to take place. We can also talk about the membranes being really thin, and this allows a short diffusion path. It means that your diffusion will happen much quicker. In animals, we can talk about having an efficient blood supply, and that helps to maintain a concentration gradient. If you can have a big difference in concentration, this means your rate of diffusion can occur much quicker. Having an efficient blood supply means that substances can be moved away quickly and maintain that concentration gradient. In the lungs specifically, we can talk about them being well ventilated, so having lots of air, and that again will help to maintain the concentration gradient. So all of these things are things that we can talk about in terms of how these places are adapted for efficient exchange of materials. Okay, next up is stem cells. So a stem cell is an undifferentiated cell of an organism, and that is capable of giving rise to more stem cells, or it's capable of giving rise to other cells, and that can arise from differentiation into specialized cells. Now, these are stem cells and they are found in embryos, and these are the really early stage of development. We can also find stem cells though in adults, and we would find those in the bone marrow. Now, these stem cells are unable to differentiate into all types of cell, but they can differentiate into blood cells. So the third place where we might find our stem cells is in plants, and we would find those in the meristem tissue. This is at the growing parts of our plant. And these stem cells can differentiate into any type of plant cell, and that can occur throughout the life of the plant. Now, we can talk about uses of stem cells and why that's important. We'll start by thinking about our plant. So our plant stem cells can actually be used to make sure that we can try and protect plants from extinction. So we can take rare species and we can try and clone these to try and create more plants so that they're saved from extinction. We can also take some meristem tissue from crop plants which have already got special features, so disease resistance, and we can clone those to produce large numbers of identical plants. So stem cells can be used for treatments, and there's two conditions we need to know about. We've got diabetes and paralysis. Paralysis meaning that there has been damage to the spinal cord and that there is damage to nerves there. Now, the problem with using stem cells to treat conditions like this is that quite often these stem cells have come from an embryo and they get rejected by the patient's body. So in order to try and avoid this, we have a process called therapeutic cloning. And this is where an embryo is produced with the same genes as the patient. And this means that they're way less likely to be rejected by the patient's body and therefore be better for medical treatment. So the way this would work is that we would take a cell from the patient with the desired genes, we would remove that nucleus, and we would fuse it with a denucleated egg cell. So this is an egg cell where the nucleus has been removed, and we now have a cell which can become an embryo. And this embryo has the same genes as the patient and means that we are less likely to have rejection. Now, the problem with the use of stem cells is that there are lots of reasons against using them. There are potential risks involved in terms of transfer of viral infections, and there are ethical and religious objections that people have for the use of stem cells. And the reason why is because we are using embryos to do this, and embryos are of course very, very early life forms. Okay, next focus is the heart and circulation. This is something that hasn't come up for a little while, so it's important that you try and remember some of these structures that we find inside the heart. I'm going to talk you through this in terms of how the structures are all involved in an order that makes sense in terms of blood flow. So we're going to think about the idea that blood is going from our heart to the rest of our bodies. It's being used up, the oxygen in there, the glucose in there, and then it's going to be returning back to our heart. So let's think about the blood then returning back to our heart. This blood, we would say, is deoxygenated, and it returns to the heart through this vessel here. It's called the vena cava. And this blood enters our heart through the vena cava and goes into our first chamber. This is called the right atrium. This is the chamber at the top on this side. We call it the right side. The blood then gets squeezed from the right atrium into the right ventricle. This is our second chamber of the heart that's important. The blood is then pumped out of this right ventricle and goes through the pulmonary artery. Now, this pulmonary artery is going to take this blood to the lungs, and this is where the blood is going to become oxygenated. So it goes to the lungs, becomes oxygenated, and it comes back to the heart. And it comes back to the heart through the pulmonary vein. Once it reaches our heart, it's now into our third chamber, the left atrium. Again, we're going to push this blood into the ventricle, this time into the left ventricle, and now we're ready for our final push out to the rest of the body. This is now oxygenated blood, and that gets pushed out of our heart through the aorta. Okay, you could be asked about how the heart goes wrong, and we're going to talk about that now. So what you'll notice in this diagram on the left-hand side is that there is something labelled there called valves. We can see lots of valves in our heart, and these are really important because they prevent the backflow of blood. If we have leaky valves or faulty valves, it means that your heart isn't very efficient at pumping blood. It means that when it squeezes, the walls of your heart squeeze, the blood isn't being forced through the right vessels, and it may end up going back into different chambers. So we're going to fix this, and you can do that through mechanical valves or through biological valves. Next thing that could go wrong is actually something called coronary artery disease. The coronary arteries are the arteries that supply the heart muscle itself with blood. And it needs that because the heart is a muscle, it's going to be respiring, and therefore it will need its own supply of oxygen. So we need to ensure that there is a really good ability of our heart to actually get its own oxygen. And the problem that we can have with coronary artery disease is that we can get something called a blockage. And this is where layers of fatty materials build up inside these coronary arteries, and it means that our blood is unable to flow through. Now, we can actually fix this. We could use drugs, or we could use a surgical procedure. So here's an example of a surgical procedure called a stent. And this is something that gets put inside of the coronary artery to try and ensure that the blockage isn't going to affect our passage and direction of our blood flow. Okay, next up is response to exercise. So you need to know about how your body is able to respond to the changes that will occur when you exercise. Now, during exercise, your muscles will be respiring at a much greater rate. And that's because the muscles themselves, the cells, are going to need to release energy for contraction. Now, muscles are adapted, they have lots and lots of mitochondria, and they are going to be increasing their rate of respiration in here. And therefore, we need more oxygen and glucose being delivered to these muscle cells. So during exercise, we need to understand this key principle, which is about respiration. Glucose and oxygen being required for this and creating these two products, carbon dioxide and water. So the rate of respiration is going to increase in your muscle cells. That allows contraction. And therefore, there's going to be an increased demand for oxygen and glucose to try and reach our muscle cells. So in response to this, your heart rate will increase. That means your blood will be pumped faster, and the breathing rate and depth will increase to allow more oxygen to enter the lungs and then be delivered to those muscle cells in the blood. It's likely that if you're asked about response to exercise, that they could present you with something like this, which is a graph showing us this response. And this was about 2 students doing some exercise and their breathing rate being measured. There are a series of questions here that I'm just going to talk you through and how you would unpick this from the graph and how you would explain some of these. So number 1 says, what time did the student start exercising? For this, we need to start thinking about where that breathing rate has started to increase. So you would simply look at your graph, you would have a look at where that breathing rate was beginning to increase and trace it down. So you can see here that there is an initial increase, and you can see here that there is an initial increase. We trace that down and we can spot 4. So this is when our students were starting to exercise. It then says describe the differences between our student X and student Y. Well, we can make a comparison in terms of their initial breathing rate being different. We can also think about the change in breathing rate that has occurred and there being a bigger increase in terms of student X compared to student Y. We can also compare how long it took before they went back to having a normal breathing rate. You can see that the exercise stopped around this time, but you can see that this took much longer to get back to the normal breathing rate compared to this student over here, student Y. So there's our differences then. Why does our breathing rate change during exercise? Breathing rate is going to increase to allow more oxygen to reach our muscle cells as there is an increased demand for this due to the increased rate of respiration. Then it says, why did the breathing rate stay high after the exercise ended? So exercise has ended here, but we need to try and remove the products of respiration out of our blood. So carbon dioxide is made during this. Carbon dioxide could be toxic or can be toxic in our blood, so we need to try and get it out. We need to get it to the lungs so that we can breathe it out. We could also have had some anaerobic respiration in here in which lactic acid is made, so we need to be able to provide oxygen to try and break down that lactic acid. And then finally, we've got give another change in the body that occurs during exercise. You could have said about an increase in the heart rate or an increase in the depth of breathing. And finally, we move to metabolism. Metabolism is encompassing a huge amount. It is all about breaking up and making molecules. So if we think about some different examples with this, we'll start with glucose. Glucose can be stored as glycogen. So glycogen is a bigger version of glucose, and this is what happens in animals. We store it as glycogen. Glucose could be stored as starch or could be stored as cellulose in a plant, so a plant would store it as starch. It could be used for cellulose, which is in the cell walls of plants. Another example is our glycerol and 3 molecules of fatty acids. Now when these come together, they will form lipids. This is another example of metabolism. We can also talk about glucose coming together with nitrate ions, and these will form amino acids. And these amino acids in turn will be used to synthesize proteins. And finally, we can talk about proteins. In our diet, we have excess. They can actually be broken down to form urea, which can then be excreted out of our bodies. So all of these are examples of metabolism. As well as these, we also have respiration as an example of metabolism. And that is us wrapped up. They are all the predictions that we have for the paper on Tuesday. We wish you the best of luck.